couple of announcements before we get started. First, guest lectures Wednesday and Friday. Wednesday is Ryan Estep, who'll be talking about herpes viruses and hopefully a little bit about cytomegalovirus, which is a really fascinating herpes virus that lots of people in the area are working on. There is guest lecture on Friday, which is George Kaysen from my lab, or who is, I should say, rather than which is. And he'll be talking about single-stranded DNA viruses, including some of the ones that he's working on in my lab. And we'll touch really briefly on some of the single-stranded DNA viruses today. Yes?
Will you post their slides?
Yes, I will try, assuming that they give them to me. I will definitely try and post, particularly before lectures. So I asked Dr. Estep to do that already, to send me his before presentation, and then pre-Dr. Kaysen, I will also ask to post those appropriately. Whether they will be recorded, I don't know. I will ask them if they're happy with doing it, and if they are, we'll have, just like we did with Dr. Hirsch. So any other questions about those things? Yes, they'll be on the exam, which is like way too soon. I agree.
Who would ask something like that?
Who would ask something like that? Yeah, no idea. So yeah, today, again, we'll talk about more of these DNA viruses, particularly the adenovirus. And sort of the big picture with adenovirus is it's got this amazing shape, of course. It's, bigger, fatter, uglier, depends on your point of view. One of these double-stranded DNA viruses that's infecting animals. We'll talk about larger and larger ones as we move through. I think the second to last lecture is on the viruses, so the truly giant double-stranded DNA viruses that are infecting eukaryotes. But these are sort of expanding again on this sort of theme of the papilloma and polyoma viruses, which you talked about last time. These have circular genomes. Now I've got a linear genome, which immediately provides an issue because you have to deal with the ends, and also has a much more complicated structure, and particularly much more complicated in terms of the genes. And the main sort of issue here has to do with splicing. And as everyone remembers, right, from last term, where was splicing discovered? Study of? Adenovirus, actually. So, and then a little bit about satellite viruses, but again, this is a kind of a plug for George's lecture on Friday here. Splicing, at first, interestingly enough, people thought that splicing only happened in viruses. And then, of course, later people found that practically all, certainly animal genes, and in fact most eukaryotic genes, are spliced and have alternative splicing. One of the things that was not as well realized is there's also alternative tailing that happens in adenovirus messenger RNAs. And we'll talk a little bit about that. It's particularly important for the late genes. It turns out that the late genes in adenovirus are all made from one transcript, but I think there's something like 30 different genes, products that are made actually from that single transcript. So really pretty amazing in terms of the variability that happens there. It's true with all of these DNA viruses, with maybe an exception when we get to the very end, but certainly the ones we've talked about so far. Very important that they regulate the cell cycle, because most cells are not replicating most of the time. And if you're a DNA virus, you're dependent on at least some of the DNA replication machinery and the cell cycle. So cell cycle regulation here, as we talked about quite a lot last lecture on Friday, is very important, and a lot of it has to do with our friends RB and P53. The difference here, again, mostly has to do with the topology of the genome. It's a linear genome, and because it's a linear genome, it can't replicate in the same way as you normally would. I guess maybe if you had a telomere. Don't know of any viruses that have direct telomeres on them, but these don't. They have terminal proteins, which are covalently hooked up to the ends of the genome, and that should immediately give you an idea on what those things might actually be doing. And then the genome sequence itself has inverted terminal repeats. And what the heck is an inverted terminal repeat? All that really means is on one strand of the double strand at one end of the genome is identical to the opposite strand at the other end of the genome. What does that mean? It means that they can base pair to each other, and it turns out that's absolutely critical for replication of these genomes. They can base pair with each other. So again, our sort of standard outline here, bit about disease. Actually, adenoviruses don't cause much in the way of disease. There are some exceptions to that, but it's really nowhere near as widespread as a number of the other viruses, particularly things like the rhinoviruses. One thing that I did want to mention here is this little dot at the top here, little arrow on it. This is the adeno-associated virus, satellite virus, which is in fact a single-stranded DNA virus that, again, George is going to be talking about on Friday. Otherwise, the structures of these viruses are really classic icosahedral, not quasi-equivalent because they've got different structures at the 5-fold axes of symmetry and the hexamers that go with them. We'll take a little bit closer look at that later on, and this has been where there's been some really amazing progress in the last couple of years, particularly having to do with the advances in electron microscopy in terms of structure determination. So we have some really good, very high-resolution structures from electron microscopy of these otherwise really pretty big virions here. Binding and entry is pretty straightforward. There's actually not unlike the case for lambda, where the receptor was actually named based on the virus binding to it and not on its actual real activity. The receptor for most adenoviruses, turns out there are a whole bunch of different ones, is the adenovirus receptor protein, also called Coxsackie virus. And then, again, big pictures here. Transcription. A lot of what we know and what's in textbooks about transcription is actually really about transcription of the major late promoter in adenovirus, because that's how a lot of the original work was done. Also having to do with splicing, of course. And then replication here, which is quite different, again, because we have a linear genome. Then getting out is pretty straightforward. Actually, these viruses really do seem to lyse the cell when they're being released. So it's much more like a bacteriophage kind of replication process in terms of getting outside the cell. Yeah?
You say the inverted terminal repeats. Maybe we'll get to that anyway, but they base pair with each other. Is that in the mRNA?
So yeah, we'll get to this. This is in the genome. So the inverted terminal repeats are in the genome. And as we'll see, the messenger RNAs are not of the whole genome. They're separate pieces, so those actually don't base pair with each other directly.
Terminal repeats don't get transcribed.
Those terminal repeats, if I remember correctly, they don't get transcribed, but we'll take a look at that in just a second. So again, where do these come from? Originally discovered in adenoid tissue, and that's why they're called adenoviruses. Some respiratory and eye diseases, but not a really massive number. The main case, and we'll get back to this a little bit later on, where people have seen adenovirus disease, turns out to be in military recruits, sort of base camp, is the case where a lot of adenovirus disease is happening. And in fact, there have been some not pretty good vaccines to deal with that, and we'll talk about that right at the end. People thought it was the common cold virus, and in fact, somebody I think sent me a link to a paper about the common cold virus being now used for gene therapy purposes. The people who wrote that article were A, wrong that they thought it was adenovirus, which is not a common cold virus, and B, it was all about adeno-associated virus and not about adenovirus either, but not the common cold virus, even though many people think that it is. Probably most important in terms of understanding a lot about what we do in terms of general, splicing, transcription, et cetera, and also many of these adenoviruses, partly I think because they've been studied for so long, have been used quite successfully, for gene therapy purposes. And then also, as we'll talk about right at the end, oncolytic viruses. They are tumor viruses, again, only in non-permissive cells. This should sound really familiar because that's what we were talking about on Friday. If the virus can replicate, it will make more virus and kill off that cell, so that cell can't become a tumor. If it can't replicate, in some cases it can then become a tumor. there are lots and lots and lots of different adenoviruses, and this was a more recent version, but again, the basic take-home message here is there's lots of different ones, and we'll talk about some of the best studied adenovirus type 2 for the most part, but again, many, many, many different ones of these. And as we've talked about a little bit before, serotypes, this is just the different antibodies. And different antibodies means you're going to have a different surface antigen on the outside that the antibodies are going to react with. And then of these, they're split up. We talk a little bit about some of the D adenoviruses, type D adenoviruses. That's this different subgroup, which have slightly different structures to them. And we'll talk about that in just a second. The structure, again, more complicated. The actual structural proteins, there are 11 of them, fit into this pseudo T equals 25 icosahedral structure. What does pseudo mean? It's made up of different proteins, and it's pretty obvious what those different proteins are if you look here at the five-fold axes of symmetry, because they have a spike coming off at each of these five-fold axes of symmetry. None of the hexameric structures in between there have this spike that's sticking out of them. Each of the hexons, and again I should have brought my soccer ball with me, each of the hexagonal units is called the hexon, conveniently enough, although more confusing, it's actually a trimer rather than a hexamer of different proteins. So the trimer is right here. Red, white, and blue, appropriately patriotic here, or bleu, blanc, rouge, depending on your point of view. Each of these wrap around each other, has these two beta barrels down here at the bottom, and then these projecting domains that come out here at the top. Again, this beta barrel structure is extremely common in different viruses. Just seems to be a good way of putting together your proteins in order to make that structure. May or may not say something about the evolution of virus structures. We can talk more about that later. So that's the hexon protein. You need many, many, many copies of the hexon protein, and we'll get back to that a little bit later on. You need a lot of copies of the protein, means you need a lot of copies of the messenger RNA. The penton protein, again, five-fold axis of symmetry. It's actually made up of three different proteins. There's the penton base, there's the fiber, and there's a knob out at the end of these. And of course, as always with icosahedral structures, you've got 12 of these. This is the version, actually the colored version of what we have in our textbook. This is one of the higher resolution structures. This came out about 7 years ago. Vijay Reddy at the Scripps Research Institute did this really nice, this is actually an X-ray crystal structure, looking at one of these adenovirus structures. So you can see the knob protein here in orange, the base in purple, and then each of the hexon proteins that are spread out relative to that. You can also, if you wanted to figure out the T number, just in case someone were going to ask you something like that, you would find each of these pink units. These are your five-fold axis, and then you just step from one to the other. 1, 2, 3, 4, 5. No movement in another direction. So H is 5, K is 0. 5 squared is 25. So it's your T equals 25 arrangement in terms of how these are all put together. Turns out these structures of the hexons are not exactly the same. That's why they're in slightly different colors here as they put them together. This is, interestingly enough, a study that was done simultaneously and published actually at exactly the same time, now using electron microscopy to get the same kind of structure. And one of the things that was really nice about this structure is not only do you have the pentons, which you can see right here, and each of those hexons, which you can see here actually better in this structure than the previous one. They're trimeric. Each of those, there's 3 little red dots. It's a trimer, 3 orange dots, 3 orange dots, 3 red dots before you get to this five-fold axis of symmetry. But there are also lots of proteins that are in between each of these hexons, just sort of providing the glue that's sticking all of these things together in this really quite complex structure. This was the case for the type 5 adenine, adenovirus type 5. Even more recently, this is literally a couple of months ago, also Vijay Reddy's lab published another cryo-EM structure that's even higher resolution than the previous cryo-EM structure. Any of you who went to Dr. Wiesler's seminar a couple of weeks ago heard about some of the progress that's been made in detectors, and basically taking a video of the electron micrograph and then using those videos to get a much higher resolution structure. And so that's what they used here. And again, some of the important aspects of these are these glue proteins. A little hard to see here, the yellow one here, the orange one here as well. And this is a different one of those. All of those previous ones were one class. This is, in fact, the class that people are trying to use as oncolytic viruses. They haven't worked terribly well. This new class that they're working on, this D class of viruses, may end up working better for these anti-tumor agents, which is one of the reasons that they were interested in this structure. And I love these structures so much that I want to show you the movie. So we'll take a quick look at the movie here of these structures. Oh, this is, let's see, where is it? This is the movie of this. This is actually the crystal structure of adenovirus 5. Again, 5-fold axes of symmetry here. Here's each of your hexons, and then the purple here are all of the structures which are linking each of these guys together and literally gluing that whole structure together. If you've got a complicated, icosahedral head-like structure like this, what do you normally need? At least what have we seen before in complicated icosahedral head structures? What do you need to put these together? Scaffolding proteins, exactly. So not surprisingly, you also need scaffolding proteins for putting together these adenoviruses as well. Yeah, sir. Sorry, was there a question? All right. So that's it for structure. But because this structure is so beautiful, a number of people have taken advantage of it, used it for art purposes. This is a really nice finial. It's hard to see over here. This is at Cold Spring Harbor Lab, where many of the discoveries were made in terms of adenovirus transcription and splicing. This was an interesting project that they put together. This is actually more about the architecture of this neat gazebo, which is actually right on top of their wastewater treatment plant at Cold Spring Harbor. And partly because they have a big issue with wastewater treatment here because they use lots of radioactivity. And so you want to make sure that you're not dumping a whole bunch of radioactivity into Long Island Sound, which is right next to Cold Spring Harbor here. So let's look at this in more of a cartoon form. These are way too many proteins for anybody to remember, including me, so they won't be on exams. And they also have this ridiculously confusing numbering pattern, as well as calling them hexons and pentons. But I did want to concentrate on a few of them. So here, hexon, again, forget what these numbers are. Hexon protein, this is the major structural protein present here on the outside. Then you also have the penton, first the base, the fiber, and the knob, and then lots of these glue proteins in between all the rest of these. And so those are the ones that these higher resolution structures allowed us to get a handle on and are probably important for some of the differences between why these things work well as gene therapy or oncolytic viruses and why some of them don't. On the inside of the particle, you have your double-stranded DNA. It's associated with lots of DNA binding proteins that bind pretty non-specifically to it. But most importantly, as far as we're concerned, you can tell because it's got this little red circle around it, is the protein which is covalently bound to one end of each of the DNA, and that's bound particularly to that genome. What does that look like? here it is, 5' end, you have a protein bound to your genome, which is, depending on the adenovirus, about 35,000 base pairs. And it has these inverted terminal repeat structures. And so just the beginning of this inverted terminal repeat is here. Here we have CAT, CATCA. Can't believe this typo. They spend all this time in the textbook talking about the inverted terminal repeats. And then if you go over here, opposite strand, CAT, CATCA. It's like, wait a minute, you guys forgot this T. How could you? But that's the idea. So inverted terminal repeat, same sequence on one strand, same sequence on the opposite strand at the opposite end of the genome. And again, always talking from 5' to 3'. And at the 5' end, you have this terminal protein, again, creatively named protein here, bound at the 5' end. Now, why do you think it might be bound to the 5' end? Could be a primer. Yes, so it's providing the OH that the DNA polymerase needs to extend through the rest of the genome. So, what other viruses do we know that need 3' OHs that come from proteins? Let's ask a quicker question about it.
I wasn't ready.
Oh no, shock horror. Oh, and actually, come to think of it, I should probably actually start the clicker program. That might help. So, gives you a little bit more time to think about it. Okay, now, here we go. The primer for genome replication in adenovirus is most similar to the primer used for replication in 5X174, SARS, polio, Ebola, or bacteriophage T7. It's the last click that matters. Oh, yes. Feel free to change your answer. Talk to your friends. See if you believe them. May have been right. These are the kinds of questions that you probably should sound familiar from some of the midterms, quite likely to be something you could see on a final. So, 10. Are there only 37 people here?
Final isn't cumulative,
but well, I guess I'm not competing well with the sun. Can't say I blame people here. So, what's the primer for 5X174? Cellular primase makes it. For SARS, seems to be the polymerase that makes it. For Ebola, polymerase makes it. For T7, probably actually the RNA polymerase, interestingly enough, rather than a primase. What about polio?
yes.
Yes. What's it called?
VPG.
VPG, exactly. Okay, any questions? Sorry, any questions about this? No, okay. So one thing that I keep forgetting to do when we're going through some of these different viruses is to include this slide, which we had way back when, the beginning, when we were talking about entry. This is a nice way of looking at how various different viruses enter the cell. So for instance, influenza, those orthomyxoviruses come into the endosome, change in pH. The M1 protein is important for getting the ribonuclear protein particles inside the nucleus. SV40 comes in through these caveolae, ER, and then the virion itself. This is a little controversial whether virion actually comes inside the nucleus or whether it disassembles right at the nuclear membrane. On the other hand, for adenovirus, it's pretty clear that you have binding by the receptor, which we'll take a look at in just a second here. That then comes into the endosome, but then the virion, at least most of the virion, escapes from the endosome and gets moved to the nucleus through normal microtubule trafficking processes, through dynein, all of these motor proteins that literally move things around here. And then at the nuclear membrane, that's when the genome is actually released, and then in the nucleus ends up being replicated. So this is the entry process. How does it get in? First, it binds to the Coxsackie and adenovirus receptor. That's the knob on the very end of each of those five-fold axes of symmetry. But it doesn't seem to be just the knob protein. You also have interactions with the base, and these are integrin proteins. These are the proteins we've talked about before, particularly if the papillomaviruses, where you have a immunoglobulin-like protein. What do these do? They love to stick to things that will then stick to this penton and bring it into the endosome with pretty normal receptor-mediated endocytosis. Now, the big difference here, get to that, is that we have viral proteases that now chop off this knob and spike protein and then also allow it to escape from the endosome. Yeah?
Just clarify, Coxsackie and adenovirus receptor?
Coxsackie and adenovirus receptor, exactly.
What chops off the
So it's a viral protease. So there are viral proteases that are involved now in entry. And so we've talked a bunch about viral proteins that are being involved in getting fusion proteins ready. Now these are viral proteases that are being used to bring the actual virion inside the cell. And then there's a different viral protease that actually breaks down the rest of the capsid at the nuclear membrane. So you have all of these proteases, and just like the case for all of our negative strand RNA viruses, nothing has been transcribed or translated yet. So all of these things have to be present in the virion in order to get this regulated degradation that takes place in order just to be able to release the genome at all. Cartoon of how these interact with each other. Again, getting back to your question about the receptor. Here we have they call the CAR protein. That's your cellular receptor here. Interacts with the knob at the end of the fiber at each of the five-fold axes of symmetry. And then the integrins here, which are these black blobs, are going to interact with the base. And it's these together which allow you to have this receptor-mediated endocytosis process that brings the virion inside the cell. Once it gets inside the cell, again, there's proteases that will chop off these bits and then allow this virion to escape from the endosome, gets transported again along the microtubules to the nucleus, releases its genome, and then that genome gets translated. Now this is transcribed, I should say. Transcription. Again, about 35,000 base pairs. We're going to spend most of our time talking about the one big transcript, which is all of these orange ones here. But there are other promoters that are present in the genome. And as usual, we've got early and late. Again, pretty classic kinds of expression patterns. Earlies are going to be important for what? Replication and messing around with the host. Host defenses is one thing. But again, this is a DNA virus. So what does it need to do? DNA viruses have a problem. Most cells are not replicating most of the time. What does it need to do? It needs to tweak the cell cycle. So also early proteins are very involved in that process. There are also some small RNAs which are made here. No one thought that small RNAs were important, when they first started studying adenovirus, but of course in the last few years it's become really important that they're what's going on here. So let's look at some of these separately. The main ones are going to be our earlies and our lates. Earlies, again, ridiculously creatively named, E1, E2, etc. E1A and E1B are the ones which are most important for regulating the cell cycle, and we'll take a look at those in more detail here in just a second. All are getting alternatively spliced to give you lots of different RNAs, but these guys basically serve the same role, E1A and E1B. E2s, on the other hand, are now the ones which are most important for replication. So E2A, DNA binding proteins, E2B, terminal protein, which is the primer, and DNA polymerase, etc. E4, we'll get to a little bit later on. Actually, it turns out that E4, even though it's a early transcript, is important for very late stages of replication here. Then we have these little ones that we're not going to bother about too much. Again, they're short RNAs. And then the late genes are all made from one promoter, known as the major late promoter. Again, surprise, surprise. All starts right here. We have a leader sequence. This leader sequence is attached to all of these other RNAs here, which are coding for classic late proteins, like the fiber protein, the hexon protein, the penton protein, etc. You'll note that they all have different ends to them here, and that's due to different tailing, which we'll take a look at here in just a second. And then we've got these short RNAs. But important as far as we're concerned are going to be these early genes here, E1A, E1B, the major late promoter, and these E2 proteins. But what about this, tripartite leader and all of these other things here? Now, you remember what gene did I say was probably going to be transcribed the most? That was like 10 minutes ago, 15 minutes ago. It's sunny outside. My brain's not working. It's Monday morning. Hexon, major capsid protein. So because of that, you have very large amounts of the messenger RNA for the major capsid protein. And this is how people discovered that splicing was taking place. And in fact, it wasn't just the hexon protein. People were also looking at some of the other structural protein genes. And what they did is they used this technique called the R-loop technique. Anyone remember what R-loops are? Yes, no? I see some nodding of heads. The idea of an R-loop is you take RNA and you hybridize it to DNA. And so you start out with a genome, in this case, adenovirus genome, or parts of the adenovirus genome. And one of the really critical aspects about figuring this out was how to get parts of the adenovirus genome. Any idea how you would get a part of an adenovirus genome? Cut it. How would you cut it? I wouldn't use a protease. I would use a endonuclease, and particularly a restriction endonuclease, right? Because those cut at specific sequences to chomp things apart. Well, one of the main people, I should say, to work on restriction endonuclease was this guy by the name of Rich Roberts, who also founded a company called New England Biolabs because they were so useful for everyone else to study all of these things. And so we still get lots of our restriction endonucleases as well. So he chopped up parts of the genome, then did these R-loop assays with them. He knew exactly how big each of these pieces were, and he could say, oh, this is really bizarre. I've got this RNA, but it's binding to different pieces that don't belong in the right place in our genome. At just about the same time, Phil Sharp, who was at MIT, Rich Roberts, by the way, was at that place where they have the water treatment plant with the adenovirus thing sticking on top of it at Cold Spring Harbor, where he was doing these studies. Phil Sharp was just up the road a little bit anyway at MIT. They had figured out a way to purify messenger RNAs. How do they purify messenger RNAs? How do you purify messenger RNAs these days? Poly A tails. and using a poly T to bind to those and then use that to pull out your individual messenger RNAs. What was the most prevalent messenger RNA in an adenovirus-infected cell? The hexon. So they took hexon RNA and hybridized it to, in this case, a EcoR1 fragment of the adenovirus genome. And basically, what they were looking for was where the RNA bound to this piece of DNA. And then the big surprise were all these extra loops that were present between different pieces of RNA, DNA that was being looped out, here A, B, and C, and then at the very far end of your hexon messenger RNA, not surprisingly, you had this little 3' tail. What was that 3' tail? What would that be? What did they use to purify this messenger RNA with the first place? Poly A. Yeah, so poly A is not in the genome. The poly A is not in the genome of the virus. So we're looking at the various different colors here, which are all DNA, which is the DNA of the virus, hybridized to the RNA from the hexon protein. Yeah?
Just going to say, a lot of times I think I know, I'm just really scared. So, yeah,
Oh,
that's it.
okay, that was it. Okay, so what we're looking at again is this is the hexon RNA bound to the DNA with, again, this poly A tail hanging off here at the end, and then hybridized to these pieces right here. And because Rich Roberts had developed all these nice restriction endonucleases, so they knew what size each piece was, they could say, wait a minute, so this RNA is binding to this piece of DNA, this piece of DNA, this piece of DNA, and that piece of DNA. That's bizarre. What's going on here? And so they came up with the whole idea of splicing, that the RNA was composed of only a small piece of the DNA that they started with. And again, everybody thought, oh, this is just some bizarre thing that viruses do. Actually, adenovirus does a really good job and splices all kinds of genes, probably way more than you would have in a normal cellular splicing reaction. But turns out this happens in cells too. Yeah?
This is all nucleic acids, though, that's doing the splicing, right?
Okay, so in this case here, this is literally purified DNA and purified RNA that they're mixing together.
But they're all nucleic acids. What is it that acts as the 3' OH to actually splice these?
Okay, so it turns out that I guess the question is, what's the mechanism of splicing? They had absolutely no idea at this time. This was in the 70s when they did these original experiments. It was only later they found that it was the whole spliceosome machinery. This is completely normal cellular splicing that's going on here. It's all happening in the nucleus with all of the regular kinds of mRNA processing enzymes.
So they can only identify where it would be spliced, but splicing can't take place by itself based on this data?
Yeah, this is ipso post facto. So you've got your DNA, you can isolate lots of genome. It's one of the reasons to work with these viruses. You can get lots of really homogeneous DNA, and then you can get the RNA, again, because the hexon RNA, because you can get lots of that. It's the one that's the most there. And then you purify each of these away from all the proteins and then just mix them back together and see where they hybridize to each other. And so that's where this is. So yeah, they had no idea what the mechanism was at this point. And it turns out this is all normal cellular splicing. It was well after this that they then discovered that RNA could actually have splicing activity. Again, completely different story. These are all spliced by the normal splicing machinery. You know, regular lariat structures, you name it, U1, U2, U3, et cetera. Yeah?
Have they ever found a virus that encodes its own RNPs, or at least the RNA part?
So the question is have people found viruses that encode their own splicing machinery? To my knowledge, no, but as they always say, ignorance is infinite.
Asking because
Yeah.
that would just mean that they would have had to have evolved, behind, eukaryotic evolution of splicing.
Yeah, and so in this case, clearly it's a case where these viruses are clearly highly dependent on their host machinery as far as that's concerned. We'll see, and actually Dr. Estep will talk about this a little bit on Wednesday. There are some viruses that do very, very little splicing. And so that's probably an interesting aspect in terms of the evolutionary things. Yeah?
Group 1 and Group 2 are
Self-splicing introns, correct. Group 1 and group 2, yeah.
So would that be applicable to what you were saying now?
So, I'm trying to not go too far down that rabbit hole here. But in terms of type 1 and type 2 self-splicing introns, so
It would still encode to have its own splicing take place, right?
right, and so if we're talking about splicing machinery, I think what Ian was asking about was more like the SNRNPs, general splicing machinery that does that. There are a few very well-known self-splicing introns present in virus genomes. And if I remember to talk about it, I will mention it when I talk about the algoviruses a little bit later on. But yes, there definitely are self-splicing introns that you'll find in virus genomes. But in terms of the sort of classic SNRNPs, I do not know of any, but that doesn't mean that there aren't any. Okay, so now we're good with splicing. Yes? No? Remember it from last term or not, as the case may be. Hopefully, type 1, type 2 self-splicing introns mean things to people. Good. Okay, so let's talk about what you get after you've done all of this alternative splicing. And we'll start at the beginning, the early genes, E1A. And as I mentioned before, E1 and E1A, E1B are the ones that are really most important for regulating the cell cycle. And this should, again, look really familiar based on what we talked about last time on Friday, and everyone's forgotten because it was a long weekend. Well, not a long weekend, felt like it because there's actually some sun shining. These are proteins that now interact with the regulators of the cell cycle, and in particular, RB. So the retinoblastoma protein, which is critical for usually shutting down the cell cycle, but activating the cell cycle once it becomes inactivated or removed from the DNA. These proteins are also very important for transcriptional regulation. They interact with the TATA binding protein, which helps get transcriptional activation and helping get transcriptional activation to viral promoters. Also interact with histone acetyltransferases. That's what this P300 is. Histone acetyltransferases do what? Transfer acetyl groups to histones, exactly, which does what? That opens up the chromatin and allows more transcription to take place. But wait a minute. These adenovirus genes aren't packed in chromatin. That was the papilloma and polyomaviruses. What are these involved in? And probably the answer is a lot like what's going on with RB, is they're also interacting with these histone deacetylases in order to get them away from not just the viral genome, because again, that doesn't have histones on it, but these viruses are all dependent on host genes. And so those host genes also have to have chromatin remodeling in order to take place there. So the cartoon of this is RB interacting with our histone deacetylase complexes, also interact with these E2Fs. Okay, what the heck is an E2F? E2F sounds like it should be a viral protein. It's not. These are cellular proteins. But how were they found? They were found in studies of adenovirus infection. So some of the most important proteins for regulation of the cell cycle were found in the study of adenovirus. So these E2 factors activate S-phase, etc. Genes. And it turns out the virus also uses them to activate its own E2 genes. But in a normal cell cycle, is what we have up here at the top, these E2 factors are just leading to expression of your S-phase proteins that you need. Again, your DNA polymerases, your primases, your ribonucleotide reductases, et cetera, everything that you would need for a normal DNA replication. How do you get these? Cyclin-dependent kinases in the cell cycle. What do? When you're supposed to be going through the cell cycle, will phosphorylate RB. That will no longer bind to these E2 factors. You'll go into S-phase. E1A binds to RB and basically pretends that it's phosphorylation. Once E1A is bound to RB, these E2 factors will go ahead and transcribe the cellular genes that are important for going into S phase, but also the E2 genes. What the heck were those E2 genes again? DNA polymerase, single-stranded binding protein, and terminal protein. Again, all required for adenovirus replication. Yeah?
Would it be fair to say that E1A is analogous to E7 and large T antigen, or maybe just E7 from papillomavirus?
I would actually flip that on its head and say for the polyomaviruses, which is where the T antigens are from, it's much more analogous to the T antigen in terms of having this transcriptional regulation and stimulation of the cell cycle. Much more like the T antigen rather than some of the other ones. Or E2 that you see in the papillomaviruses. Yeah, E1 and E2. But here, now, the other thing that E1A interacts with is P53. So again, we've got these two tumor suppressor proteins, cellular tumor suppressor proteins, retinoblastoma and P53. E1A regulates P53, but in actually kind of interesting ways. Basically, presence of E1A leads to lots of P53 being around. And that actually seems to be kind of counterproductive, and we'll see that in just a second here. But E1A, again, is important for getting rid of RB, but it's also important as a transcriptional regulator for its own proteins and some other cellular proteins. One of those is this P14-ARF protein, which binds to MDM2 and gets rid of it. Okay, what's MDM2 normally do? The normal process of MDM2 is to bind to P53 and lead to degradation. So if you get rid of MDM2, you end up with a lot of P53. P53 is usually self-regulating because P53 is also a transcriptional activator. It turns on MDM2. MDM2 will then bind to P53 and lead to P53 degradation. E1A, and this is the version in the textbook which really confuses the heck out of me, is it's blocking P53 activation. It's actually blocking the transcription of MDM2. So not just so much the activation of P53, it's blocking MDM2. At least it makes it easier for me to understand this. So you're blocking MDM2 either directly through E1A or indirectly through P14-ARF. And what this means is you've got a whole bunch of P53. Well, what does P53 normally do if you've got a whole bunch of it? Leads to apoptosis, which generally is not a good idea if you're a virus that's dependent on a host being viable and replicating all of its genome, et cetera, in order to make more virions so you can go out and infect something else. However, P53 is also good at expressing a number of other proteins that might actually turn out to be useful. So E1A by itself is actually turning up P53, probably a pretty bad idea in terms of apoptosis and killing things off. We'll see why that might turn out to be very useful a little bit later on from our point of view. In terms of the virus's point of view, there's also E1B. E1B basically says, oh, we've got all this activity of P53, which is going to lead to apoptosis. This is a bad thing as far as I'm concerned as the virus. Again, to totally over-anthropomorphize here. We want to shut down apoptosis. And how do we do that? We interact with the proteins that are involved specifically in this apoptotic process. BAX, BCL2, et cetera, normal apoptotic pathways. So the E1B really serves to block apoptosis. So E1A turns up P53, E1B shuts down the apoptotic pathway. This seems really backwards in terms of having virus replication take place, but it's, adenoviruses, it seems to work reasonably well for them. So this is the regulation of the cell cycle. First, get rid of RB, and that way you're stimulating the cell cycle. That's what E1A does, but at the same time, it's turning on P53, which would normally lead to apoptosis. However, it's also making E1B, which then will shut down that apoptotic process. Yes, no, huh? Okay, so now we need to replicate our genome. How do you replicate the genome? Well, as we already mentioned, the terminal protein is bound to the 5' end, and it serves as a primer for your replication of the genome. So here, terminal protein is here. It's got an OH. OH is on a serine, slightly different than the tyrosine that we had before. Doesn't really matter. That then serves as the primer for the viral DNA polymerase. The viral DNA polymerase starts to replicate the genome, and then there's a single-stranded DNA binding protein. This is a viral-specific single-stranded DNA binding protein. It's an incredibly cooperative protein. And so as soon as you have the two strands come apart, one of those is going to be, of course, a single strand. That gets bound to by this single-stranded DNA binding protein. As soon as you have one of those single-stranded binding proteins that binds to it, another one's going to bind right next to it, and then next to it, and next to it, and next to it. So basically, this single-stranded binding protein serves as a helicase. So there's no specific helicase in any of these adenovirus replications. It's just the single-stranded binding protein. And the fact that this single-stranded binding protein can bind so cooperatively that it pulls the two strands apart. So just a single-stranded binding protein. And then you, if you're the DNA polymerase, again, you've got your primer, you've got your template, which is now single-stranded because it's a single-stranded DNA binding protein, goes down to the opposite end of the genome, falls off, and could go back and do this again. This works, but you're only making one copy, and this extra, what's happening with this extra strand, which is flopping out in the breeze here? Well, now we come back to those inverted terminal repeat sequences. So you remember the sequence here on this strand is the same as the sequence here on this strand, but that also means that this end is complementary to that end. So what can happen is this single strand forms the frying pan, as they call it, about 100 bases here. Now you've got a double-stranded piece of DNA that serves as the binding site for your terminal protein and the DNA polymerase. It can now replicate around this part of the genome as well. So you've copied this strand, and then you copy this strand. So it's not a simultaneous. It's not like you have with leading strands and lagging strands. It's always just one strand, the leading strand, as it were, being replicated in one fell swoop, starting at one end, going all the way down to the other end. And it's just because these two ends are complementary to each other that it can give you this extra piece. Okay, so it's all about the inverted terminal repeat sequences. We haven't talked about the rest of how you get the rest of the late proteins. So again, early proteins are involved in replicating your genome. Late proteins are all of your structural proteins. Those structural proteins are made not only by differential splicing, but also differential tailing. And so these are 5 different sets of genes which are made first through splicing, but then, because transcription and mRNA modifications are all happening co-transcriptionally, you have splicing that takes place, and then tailing, i.e. cutting of that messenger RNA, poly-A tail addition, that can happen in 5 different places. And so here, these are your 5 different classes of late genes, and each of those then get spliced into different pieces. And this is how you can have, actually it's almost 30 proteins made from one original transcript. So this splicing, the splicing here at the beginning, these splicing which Roberts and Sharp saw, they also got the Nobel Prize for this, those are always at the 5' end of your messenger RNA. The 3' end is completely variable due to different splicing and different tailing that takes place. So it's both splicing and tailing which is involved in regulating which messenger RNAs you get. Okay, how do we get out of the cell? These are the early proteins, which are serving as a late role. It's very confusing. They're being made early on. Probably the reason for this is that you have to have accumulation of these proteins and accumulation of the messenger RNA before they can actually serve their role. So two of these, E3 and E4. E3 seems to be the death protein, kills off the cell. And E4 in particular is really good at killing cancer cells as well as killing the cells that were producing the virus. So again, the idea here is that you're going to lyse the cell. You're going to completely lyse the cell after you're done replicating and making a whole bunch of virions. So in order to get out of the cell, it actually turns out that apoptosis might actually be good, but only at the very end of the virion replication process. So it seems that E3 and E4 are kind of now counteracting what you had with E1A and E1B. So E1A and E1B, we're saying, okay, let's not do apoptosis now, but now that we've replicated our whole genome, made all of these late proteins, had them all fit together into virions, now apoptosis is not such a bad idea because the cell's going to die. As the cell dies, everything gets broken down, including all the membranes. Now the virions can get out and go and infect some other cells. Yeah?
So instead of just allowing this huge accumulation of P53 to now initiate apoptosis, they have their own proteins that now initiate apoptosis?
That seems to be the case, is that you actually have your own proteins that are involved in stimulating apoptosis. So it's not the P53 driven, it's virally driven. Although there may be some interaction with the P53 there as well. So partly because these viruses are doing this really bizarre thing in terms of apoptosis and all these different kinds of regulations, and we've been working on adenovirus for so many years, people have now tried to take advantage of these viruses to now use them for our own purposes. And a couple of these are tumor therapy and various gene therapy applications. So we'll start out with the gene therapy and also being used for vaccine. These viruses are really good at getting inside cells and then getting whatever's inside your virion to the nucleus, right? Because they have the breakdown in the endosome, and then that whole thing gets transported to the nucleus. The viral protease breaks down the capsid, and you get into the virion, you get into the nucleus. That's exactly what you want to do with gene therapy, right? Is you want to get that gene, whatever's in the inside there, into the nucleus. And so you, however, don't want to be killing off the cell, and particularly don't want to be stimulating the cell cycle. Well, if you don't want to stimulate the cell cycle, get rid of E1A. And actually E2A as well, but the main thing here is just getting rid of E1A. So you get rid of E1A, you don't stimulate the cell cycle, but you do get really good gene delivery to the nucleus. So that's one thing, and in fact, a lot of the gene therapy trials right now, I think something like 20% of gene therapy trials use adenovirus and some kind of modified adenovirus in order to try and get those new genes inside the cell. On the other hand, people are now taking advantage of the fact that there's this really bizarre mechanism of turning up P53, which then is potentially a way that you can now have these viruses specifically kill tumor cells. And so the idea here is you want to be sure that the normal cells that end up being infected by these adenoviruses don't get killed by the adenovirus, all these late proteins going to apoptosis, but the cancer cells will get destroyed in this particular process. And the main way that this is being done is to make an adenovirus that's missing E1B. Now, what does E1B do? Blocks apoptosis by expression of P53, right? So the vast majority of tumor cells don't have P53 in them. So if they don't have P53 in them, then the adenovirus lacking E1B will replicate and make more virus and kill off that tumor cell. But if it has p53 in it, what's going to happen? That particular cell will undergo apoptosis, and so that virus is never going to replicate. So it'll be one cell. So it's kind of how you say, okay, we're going to not worry about that one cell that gets infected, or the cells right around the tumor, those that are actually p53 positive, but all the ones that are p53 negative, the virus is going to replicate like mad and blast apart those tumor cells. That's the theory. How well it works, still kind of up in the air. Was a really promising candidate called, I think, Onyx something or other, whole biopharmaceutical company. But it is, in fact, approved. Was a clinical approval for using exactly these kinds of viruses, adenovirus missing E1B for some head and neck cancers that's being used in China right now. So actually being used in the clinic.
Aren't tumor cells already mutants and don't do normal apoptosis and kind of just the growth just continues to proliferate anyway?
So they're not undergoing normal apoptosis, but a lot of the reasons for that is they don't have p53. So
Normal tumor cell.
tumor cells normally don't have p53.
So why would, I guess I feel like the virus is doing what the tumor cell already does. The tumor cell lacks p53, but you're telling me that this genetic therapy inserts a virus that lacks E1B,
Yeah.
normally blocks p53, so, oh wait, p53 is not already there. Right?
Yeah, so p53 is not there, which means the virus can now replicate, and it undergoes, as we were talking about before, it's the viral-dependent apoptosis now, not the p53-dependent apoptosis happening. Yeah.
It's E3 and E4 that you really want. You want to get in there because they're the ones that are stimulating the apoptosis in the cancer cell.
Yeah, excuse me, E3 and E4. But those have to then build up, and you have to have this whole replication process, which normally wouldn't happen because in the lacking E1B, you've got E1A, which is going to lead to apoptosis. Any cell which is infected by that virus is going to be gone before the virus has a chance to replicate. The idea here is that you have virus replication now going on in p53 minus cells. So the lack of E1B is just so if you happen to infect a non-cancer cell, then you don't worry about the virus replication. Yeah, you can sort of think of this like a vaccination, which is great because there's another vaccination thing that I did want to bring up here really quickly. I mentioned right at the very beginning of class that usually adenovirus disease is not a big problem, except in base camp. And it turns out that many military recruits were coming down with these, respiratory diseases, and it turns out that you can actually get vaccinations, but these vaccinations work orally, which to me is really bizarre. Is that, again, this is a respiratory disease. Normally it's, airway epithelia, but it turns out that you can take these two viruses, and that's what they are, live oral vaccines here, which don't cause much in the way of disease, but give you a really nice immune response, which will, and it's actually completely reduced the amount of adenovirus disease in base camps. So we'll stop there. Again, guest lectures on Wednesday and Friday. Highly unlikely that there will be clicker questions those days.